Okay, I think we'll go ahead and get started. I'm really delighted. We've got two of our newest assistant professors who will be telling us about their planned research, the work they've done in the past, so we can get to know them a little bit better and, really welcome them to the Microbiome Center. I know they've been interacting with many of us over the past semester, and we want to make sure they still feel connected to us, even though we're all in our little cubicles doing our own thing these days. So please continue to reach out to each other and to build collaborations. I think at least we've heard from the College of Ag that we're moving forward despite the problems of being remotely located at this point. So I hope everybody's thinking about grants and working on grants together. If there are groups that would like to organize group grants and you would like some help from the Microbiome Center, please let me know. Also, if you need letters to go with your grants saying that we will use the Microbiome Center's platform for anything, please just let me know. The other thing that I have is I have a set of professional development tools that we make available to the people in the Microbiome Center, so I'd be happy to write up something for your grant related to those broader impacts as well. So keep that in mind as you're planning your next steps. So without further ado, are there any other announcements? Okay, without further ado, I'm introducing first Sharifa Crandall, who is an assistant professor in plant pathology and environmental microbiology.
Great. Hi, everybody. Thanks so much for having me today. And I am coming to you from my cubicle that happens to be the Pennsylvania countryside. So I never thought I'd have the Susquehanna River running through my basement, but that's exciting. So. But I'm so excited to talk to you a little bit about what I've been doing before coming to Penn State. And actually my start date was February 1st, so I'm very new. And I'm also going to share with you today a little bit about my research vision here and what and some even potentials for collaboration. So today I'm going to talk to you a little bit about fungal dynamics, diversity, and function to really improve plant health. And if you want to think about my research program, it has these sort of three elements. You can also think of them as three very large stylized fungal spores. You have, I'm really interested in understanding fungal dynamics and sort of in sort of the context of epidemiology. Understanding fungal diversity, and not just, with one or two species, but with whole communities and microbiomes. And then also, I see my research program moving towards trying to understand fungal function. And this is all within the context of trying to build resilient fungal communities. Those are communities that happen to be able to deal with disturbance. And then again, this is all sort of within the broader context of plant health. So I have three major questions that sort of shape my research. And the first is, how does environment shape fungal communities and at what ecological scale? The second is, how can we actually predict plant host phenotype based on soil microbial functional traits? And then finally, what are resilient fungal microbiomes and how can they really influence plant and soil health, and how can we harness this information to really improve plant and soil health? And so first I'll talk to you a little bit about some of my previous research that I did during my dissertation work, and then also some work that I've done for my postdocs. But here, I actually worked in California, and that's a lovely picture from one of my field sites. Promise it's not stock clip art. But it's what I worked on was the question, what environmental factors really drive the dynamics and diversity of microbial fungal communities across a vegetation mosaic? And here, by vegetation mosaic, I mean different habitat types. So we have everything from coastal redwood forests to mixed evergreen forests dominated by sort of Douglas fir trees, chaparral ecosystems that are a little bit more shrub-like, and then again coastal prairies, but I actually focused on the first three here predominantly. And just for a little bit of context, the climate in California is very different than Pennsylvania. We have, you know, very little rain in the summer months in, you know, June, July, and August. And then, of course, we get rain during the wet season, which is predominantly in the winter time, and there is very little to no snow. And these are some pictures of actual fungal spores within these different habitats and environments I took pictures of under a scanning electron microscope. They're absolutely gorgeous, if you ask me. And you can see that these fungal spores happen to be the reproductive and dispersal structures for fungi, and they're what really infect plants from sort of a plant health perspective. And so what I did was actually try to answer this question about sort of spatial temporal differences by really collecting aerial and rainwater spores across the landscape. I did so with the volumetric spore trap right here, and it really leaves a little spore trace on a slide. And then I used a very high-tech spore trap, which really involves, you know, a mason jar and duct tape. The mason jar was autoclaved prior to putting it into the environment, and a funnel. And this way I was able to collect rainwater. And I was able to look at airborne and spore density and diversity by looking first under a microscope and actually counting the number of spores to get a sense for the amount of inoculum in the air, as well as taking those rainwater spore samples and collecting the DNA for them for DNA amplicon-based sequencing. And so some of the results from this study actually show that sort of regardless of where you are on the landscape, you can be in the shrubby-like habitat in chaparral, you can be in a forest, but really what's happening is that the inoculum in the air, the spore density, is really following sort of that same trend that we saw with the precipitation and relative humidity, and that really it seems like that they're really following these sort of these curves and these trends over time. And if you were to do some multivariate modeling, which is what I did, it really looked like temperature and relative humidity really seemed to be driving what we saw, and that you can also use temperature and relative humidity to really predict airborne spore density over time. So what was interesting though was that airborne spore density did not really change based on the habitat type. Like I said, it really didn't really matter where you were on the landscape. It was just the spores were kind of acting in synchrony, in unison. And so they really did seem to peak over the wet season over natural vegetation. Air temperature, relative humidity, again, were strong predictors. And overall meteorology rather than vegetation, in this case, in natural systems in California, at least along the coast, were sort of a better predictor for spore density across different timescales. You might be asking yourself, well, why is this important at all? Or, you know, how could we use these sort of baseline data? And they can really help with disease forecasting models, which are really important for plant pathologists, especially when you have forest pathologists who are interested in, say, when should they spray for different types of inoculum. They can get a sense for, okay, this is the right time of the year to do it. And that way they reduce the amount and impact of the spraying in the landscape. So again, this is sort of baseline data, but much needed. And believe it or not, we don't really have these data. We do for maybe bacteria, but we don't really for fungi. So this was kind of neat to do this study. And then moving on, I wanted to really look at sort of the diversity from those rainwater spore traps sort of across time and space. And so very quickly, I just wanted to talk a little bit about what we already know in the literature about fungi and fungal communities. And that is that fungi are ubiquitous, meaning they're on everywhere on the planet. You can find them in the Arctic, you can find them, you know, in deep sea vents. And Miles Joseph Berkeley, no, that is not Ben Franklin, but Miles Joseph Berkeley was a botanist and a good friend of Darwin's, and he wrote in a letter to Darwin, "Indeed, were not fungi so much the creatures of peculiar atmospheric conditions, there would seem to be no limit to the diffusion of their species." I think that's so cool. And basically, we know that fungi are ubiquitous, yet not every fungal taxon is everywhere. And that is important to think about. And so with the advent of high throughput sequencing, more and more studies have been coming out over the past decade or so that are allowing us to understand sort of the structure and the diversity of soil fungi across time and space. And so there was a study done by Jennifer Talbot where she and an army of undergraduates marched across the US and basically took soil samples and found that there was strong spatial structuring in the West compared to sort of the Eastern United States. You can see that. And then of course, you know, in Alaska, the fungi are always doing their own thing. It's Alaska. So what's interesting here is that, you know, they were able to see sort of this signal of different taxa sort of at the continental scale. Well, my question was, well, what about at finer spatial scales? And so I collected these rainwater spore traps more at the 5 kilometer to 10 kilometer scale, and I did so to really ask, does sporulation of the microbial taxa even change during the wet season? Do we see more earlier or later during the season? Are they different in which OTUs we find? And so this is all sort of when environmental DNA was becoming like a big thing, and it was kind of exciting to learn these techniques in the past. And so I filtered the water that I found, extracted the DNA, and in this case I was only concerned about the fungi. So I looked at the ITS region, ITS 1-2, and some of my results are actually what you would expect to find. That around almost 50% of what we saw across habitat types were just Ascomycota, and a smaller percentage of those were Basidiomycota, and this corroborates with other data from other studies that would say something very similar in other regions of the world. But what was kind of neat was when I looked at my data and I analyzed them for the most abundant fungal genera. We kind of find some of those usual suspects of plant pathogens, where you found some Alternaria, Cladosporiums, and different Fusaria. And this was actually heartening that you could actually start to tease apart, you know, what you might actually find in these particular habitats. And when it came to diversity, we actually found that there was a higher fungal diversity in the mixed evergreen forest compared to the redwood forest and the more shrub-like chaparral. And we think that might be the case because if any of you have actually gone hiking in a mixed evergreen forest, it's architecturally very complex. There are many different niches, many different understory plants, and the canopy is very varied. And so I think this is a really great sort of first pass for trying to understand sort of fungal community diversity sort of in these different vegetation types. Now, what I found to be very interesting was sort of a strong temporal structuring of the spore composition that was collected in the redwood versus chaparral versus mixed evergreen forests. And here what we find is that when I sampled across weeks 1 through 9, I did an NMDS ordination, and what you actually see here is that weeks 1 and 2 fall out kind of consistently amongst themselves. And what we see there is that you can actually think, well, maybe there's a sort of, in terms of the phenology of these communities in rainwater, that weeks 1 and 2 seem to be, the taxa are very similar compared to later in the season. And so here rainwater, and when it was actually raining during the wet season, seemed to really be driving spores. So when it rains, it's spores. And that was kind of neat to actually be able to quantify that. And we've been thinking about that a lot, and we've shown it sort of for individual species, but not on the community level, and especially in sort of these Mediterranean-like climates. And so I was also interested in sort of modeling meteorological variables onto my spore, rainwater spore data. And so what I was actually able to find here was that soil moisture and soil temperature seem to be the largest factors in terms of sort of driving the rainwater communities that we saw. And I also looked at fungal spore traits. And here I'm talking about more morphological traits. And later on, I'll talk about how I'd like to expand some of my research ideas into looking at more genetic traits as well. But here I actually looked at traits like spore width and parameter and sort of the different major and minor axes lengths. And what I found was that in the chaparral, there were significantly larger, more elongated spores compared to the forest, where you have smaller, rounder spores. And so this is an ecological result, right? And others have shown that that spores can actually have phenotypically plastic characteristics, and that perhaps in the chaparral, remember, this is all taxa blind at this point. Now I'm just looking at the actual the traits, and what you can see is that in an open-like habitat like the chaparral, you would expect to find spores that can disperse farther, that are more aerodynamic, much like a football, whereas in a forest, you'd find sort of smaller, rounder spores, much like a tennis ball. So what were my results here? Just to recap, we had strong seasonal temporal structuring of fungal communities across a vegetation mosaic, yet little spatial structure. And there was soil moisture, especially after the rain, and soil temperature, which were really driving the spore dispersal that we found. And spores were large and elongated in the drier habitat types. So next, I'd like to talk a little bit about some of my postdoctoral work that I did out in California. And I worked in two places. I worked at Cal State Monterey Bay with Dr. Timothy Miles, and I also worked with Dr. Frank Martin at the USDA ARS. And so today I'm going to talk a little bit about my microbiome project. And that project's major question was, does the root microbiome of avocado really differ in healthy versus infected roots across ecological scales? So again, it's again the same theme of looking at sort of different sites and taking different samples sort of across different scales, and then really saying, well, what's going on with the microbiome? And so I actually did it within the context now of a soilborne pathogen called Phytophthora cinnamomi. And this pathogen is pretty gnarly. If you look at these are the zoospores that are attacking a root here, and there's a lot of them. And basically, this pathogen is a real problem on different crops, on different berries, such as blueberries, on rhododendrons, again on avocados, and then on various ornamental Christmas tree species. It's also a really big problem in natural systems in Australia in the Jarrah forest. And here you can see these skeleton-like trees all throughout the landscape. And this is pretty sad, actually. And so, and I'll tell you what's sad. Look at this tree right here. This is an avocado tree, and it is experiencing branch dieback from root rot disease. And these are what the roots look like. These are healthy roots. These are roots that are exhibiting a lot of necrosis. They're really black. And so my specific questions for this particular project were, does the avocado root versus soil microbiome really differ at different spatial scales? Are P. cinnamomi infected roots significantly associated with distinct fungal and bacterial communities compared to healthier roots? And can we actually screen for secondary root pathogens that can serve as indicators of stress in avocado? And so Again, I was working in sort of this coastal band in California from Santa Barbara down to the southern coast growing region near San Diego. And in order to really get a sense of ecological scale here, we actually sampled from 18 different farms, which was really great to get like sort of a feel across the landscape. And this is what it looked like. And we sampled and had some wonderful help from some undergraduates, which was really great. And so we extracted DNA from the roots. We actually conducted qPCR, a real-time PCR, to really detect cinnamomi to make sure that indeed we were finding roots that were infected by the soilborne pathogen. We used the ATP9 NAD9 mitochondrial locus. And that locus is really great because it's species specific, and it's a locus I've worked on with Timothy Miles. And it's really great because, you know, there are other Phytophthora out there in the environment. So we wanted to make sure that we were actually finding roots that were indeed infected with P. cinnamomi. And another reason why just briefly, that this pathogen is really gnarly. It is, well, it's interesting. It has a long incubation period, maybe, you know, where the tree is asymptomatic. Sounds a little bit like COVID-19, but basically, you know, you can't tell if, you know, a farmer can't tell if their tree is infected, and they have to really dig down into the roots to find them. And even then, the necrosis doesn't really show up until like a month or 2 or 3 after infection. And by then it's almost too late. So it's really great to be able to maybe ask, are there quick ways to figure out, you know, can we actually diagnose P. cinnamomi quickly, rapidly in the environment? And secondly, can we actually also use the microbiome as sort of indicator microbes? And so what I did was, again, this was mainly amplicon-based sequencing. And I had a bioinformatics pipeline, and I'd be happy to talk to somebody over email or whatever it may be if you're interested in how I actually did it. But in the interest of time, I can't go through the gory details, but what I can show you are some of my results. So if you were to compare the bacteria and fungi inside the root, so the endophytes versus in the rhizosphere, you really see that yes, there were sort of a difference in the OTU abundance and diversity found within the roots. They were significantly different. So that's at the scale of the root. And this is to be expected. We see this in other types of crops. Not even woody crops. We see this with rice in the Central Valley, where the root endophytes versus the rhizosphere are very different. Now, if you were to actually look at now across, like all samples, doesn't matter if they're infected or not, just all the samples for the different regions, growing regions, the Central Coast versus the Southern, you could see that in fungi, there wasn't much of a difference, but bacteria were definitely different based on the coastal growing regions. And it might have to do, again, with soil amendments or different practices. And again, these are actually at different latitudes as well, the southern versus the central coast. So are the communities in the healthy versus infected roots even any different? And, you know, when it comes to the bacteria, the bacterial endophytes, no, you don't really see much structure there. But we do see with the fungi that in P. cinnamomi negative, that means more the healthy roots, where P. cinnamomi wasn't present, that the bacterial community, the fungal communities were a little bit different compared to the red dots here, which are the infected roots. And so if we were to really look at these fungal root endophytes a little bit closer and look at sort of what we can find, I would like to show you that this branch here and here, and I'm gonna zoom in here. You can see that I'd like to highlight the Nectriaceae family, where they are very much like sort of a notorious soilborne pathogen family with Fusarium, etc. And there seemed to be a higher abundance in the infected roots. These so-called pathogens still existed in the healthy roots, but there were just more of them in the infected roots. And so here is just an example of the 20 most abundant pathogens found in infected roots. And we can see we have some of the usual suspects, including those like Colototricum that cause anthracnose on avocado. And for future work, I'd really like to know, what is this phytobiome doing? So I'd like to look at functional metagenomics and screen for valence markers, perhaps for PC and secondary pathogens. I'd like to screen for suppressive microbes in the soil, and this would be a separate project. And I'd love to talk to anybody who's interested in looking at suppressive microbes in the soil and doing actually controlled experiments in more of a greenhouse setting. And then I'd also like to run field trials and experiments for chemical and biocontrol to really improve tree health for early infections and for management. And so I'd like to spend the next minute or two just talking a little bit about Penn State and my plan and vision moving forward. So I'd like to continue to work in California, but also build a really robust program here in Pennsylvania on soilborne disease management and how we can actually use fungal community ecology and microbiomes to really inform this disease management. So first, I'd like to ask, which environmental variables are the best predictors for when and where we can find soilborne fungal pathogens? Again, I'd be looking at meteorological variables, but I think this would be very cool and to run some real models with epidemiological models, possibly with Paul Esker. I'm also really excited about understanding how does soilborne fungal pathogen diversity and structure, of course, overall fungal phytobiome diversity, change at these different scales, from the root, from the plant, from a farm, and across growing regions. And it'd be very cool to do this here in Pennsylvania and to do it with other types of crops. And finally, I'd like to look at what soilborne fungal functional traits allow them to disperse and persist. And there are some microbial traits that are very important to fungi, such as timing of spore release and also important to pathology. There's also spore ornamentation that allows dispersal, lignin degradation, stress tolerance, heat tolerance, and of course, melanized sclerotia. And finally, I'd really like to work in different crop pathosystems in Pennsylvania. So if you're listening and you're out there and you're interested in collaborating, let me know. But I'd like to work with apples and peaches and apricots, still working in those sort of orchard-like systems, as well as with berries, continuing with blueberries and also with grapes. And I'd like to work on tomatoes and potatoes, and predominantly on soil-borne wilts like Fusarium, but also Verticillium as well. And of course, I'll still try to leverage the ties and collaborations I currently have in California. And for future collaborations, I'd like to continue to work as a community ecologist and to really frame my work in with theory and to really look at ideas like functional redundancy, community assembly, and priority effects, especially after soil disturbance. I'd like to look at phytobiomes within the plant health context and really connect above and below ground plant fungal response to abiotic and biotic stress, and I'd like to use metagenomics to do so. And finally, I'd like to do some more epidemiological modeling for emerging soilborne pathogens. And finally, I'd like to look at soilborne pathogen viability, and I'm very happy that I'll be working with Estelle Corradou, who you're going to hear from very shortly, on a project on this as well. And finally, I'd like to acknowledge all my amazing mentors, my funding sources, and of course, my students who are at the heart of everything I do, and whom without them, this research would not have happened. So thank you so much. I'd love to take any questions. Oops,